So now let's talk about neurotransmitters. So neurotransmitters are the chemicals that are released from a presynaptic cell that cause the postsynaptic cell to respond. So when we talk about chemical synapses, the chemicals we're talking about are the neurotransmitters. And there are many, many different kinds of neurotransmitters and many different ways of classifying neurotransmitters. And so these are just a few examples classified broadly into three groups. So the one thing to point out is that most neurons only release one kind of neurotransmitter. And so when we talk about a glutamate neuron, for example, that means a neuron that only releases glutamate as a neurotransmitter. And sometimes you'll hear the word glutamatergic, which means a neuron that releases glutamate. The exception to that, as we'll talk about more in a minute, are the neuropeptides. But broadly speaking, we can classify neurotransmitters into three groups. These three here are the amino acid neurotransmitters. So if you know what an amino acid is, it's just something with an amino group and a carboxylic acid group. And so all three of these fit that definition. So glutamate and glycine are both amino acids that are also used to make proteins. So they're found in all cells. It just so happens that in the neurons that release them, they're also packaged into vesicles and released as neurotransmitters. GABA, meanwhile, gamma aminobutyric acid, is not one of the amino acids that are used to make protein, but it's actually derived from glutamate. But its only real job is as a neurotransmitter. So again, we call these the amino acid neurotransmitters, and they differ a lot in how they affect the postsynaptic cell, but generally speaking, especially in the central nervous system, these neurotransmitters are responsible for the bulk of the sort of fastest neurotransmission synapses, and they're generally either excitatory or inhibitory, which means that they either trigger action potentials, especially in the case of glutamate, or they prevent action potentials, especially in the case of GABA and glycine. So again, glutamate is mostly excitatory, and again, we'll talk about more what that means in a second, whereas these two are mostly inhibitory. And so again, those are the three what we call amino acid neurotransmitters. The other kind of small molecule neurotransmitters are mostly lumped together in what we call the amine neurotransmitters, sometimes the biogenic amine neurotransmitters. And they're just called that because they all have at least one amino group. So like here for this choline group of acetylcholine. And so these are the amine neurotransmitters, but they don't have the carboxylic acid group, so they're not amino acids. And there are more of these, and they differ quite a bit in structure and then their role. These tend to be slower in terms of their response in the postsynaptic cell. They're not, strictly speaking, excitatory or inhibitory. They're what we often call modulatory. The exception to that would be acetylcholine. Acetylcholine is weird for a lot of reasons. Acetylcholine, we already said, is found in the motor neurons that control muscles. And so it is, for the most part, excitatory in the peripheral nervous system. But the rest of these, especially in the brain, are what we call modulatory. And I should point out, too, even the amino acid neurotransmitters like glutamate and GABA can also have modulatory effects, as we'll learn later. What matters is not so much the neurotransmitter, but the receptor. So whether or not a neuron is excitatory or inhibitory or modulatory or whatever depends on the receptor it binds to, not the actual molecule itself. And then the other major type of neurotransmitter are the peptide neurotransmitters. So the word peptide should sound familiar. It's another word that means protein, because a protein is a chain of amino acids connected by peptide bonds. So sometimes when we hear the word peptide or polypeptide, we really just mean protein. But peptide neurotransmitters are usually very short proteins or peptides. So for example, this is substance P. So substance P only has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 amino acids, which is pretty short for a protein. So most proteins elsewhere can be hundreds of amino acids long. And so these are very short. So they're released from neurons but they differ from the small molecule neurotransmitters, the amino acids and the amines, mostly in that, one, they are made differently, so we'll talk about how they're synthesized, but also they are released as co-transmitters. So that means they're almost never released by themselves. So whereas, again, with these other groups, so if you have a dopamine neuron, for example, it's going to release dopamine, but not one of the other small molecules. So for example, there's no such thing as a neuron that releases, say, dopamine and serotonin. But you might have a neuron that releases dopamine and enkephalin, or dopamine and substance P. And generally, that's true of all the peptide neurotransmitters. So peptide neurotransmitters are, as far as I know, never released by themselves from synapses. Also, the way they're released is different. So peptide neurotransmitters are often released only from a synapse if that synapse is highly active. So in other words, if you just have a weakly activated synapse, only the small molecule neurotransmitters are released, whereas the peptides are released later. So now let's talk about how neurotransmitters are made. All neurotransmitters are made in the cell that they are released from. So a neuron's job is first to synthesize its neurotransmitter and then to release it. So for peptides, the process is obviously starts in the cell body, because peptides, neurotransmitters, like all proteins, are made by ribosomes, and we've already talked about the fact that ribosomes do not exist in the axon. So there are no ribosomes in the axon or in the synaptic terminal. So peptides have to be made in the cell body. And they're mostly made on the rough endoplasmic reticulum. So remember that endoplasmic reticulum is this network of membrane in the cell body, and rough ER has ribosomes attached. And those ribosomes make proteins that are going to either end up in the membrane or going to be released from the cell. And with peptides, they're usually made as these larger precursors. So when we say precursor, it just means a larger protein that's going to be later cleaved or cut into a smaller peptide, which will actually be released. That usually happens in the Golgi apparatus. So these the rough ER will produce these little vesicles that bud off and then get absorbed into the Golgi apparatus. And as the protein passes through the Golgi apparatus, they get cleaved into the active form. So there's some cleaving. There's sometimes some other rearrangements that happen until eventually the neuropeptide gets concentrated into these big vesicles called secretory granules, or sometimes called dense core vesicles. And those get transported down the axon. And we talked all about active transport before. So we said that these secretory granules can't just wait for diffusion. There's a whole process of motor proteins that essentially carry these vesicles along the axon toward the axon terminal. Requires energy until they get to the axon terminal itself. So again, that whole process starts in the cell body, moves down the axon, and eventually makes its way to the axon terminal, where it joins the other synaptic vesicles, which are going to contain the small molecule neurotransmitters. Those, however, are mostly synthesized in the terminal itself. So at the terminal, you have precursor molecules that are converted into the neurotransmitter by the use of specific enzymes, and then after they're made, they're packaged into the vesicles by specific transporter proteins. Here you can see what those different kind of vesicles look like. Again, the just regular, what are called, sometimes just called synaptic vesicles in an electron microscope image, are these small hollow looking circles, whereas the dense core vesicles or secretory vesicles are larger and they're much darker, because those proteins are bigger, and therefore under EM they look darker or denser. So the small molecule neurotransmitters, again, they are made in the cell, mostly in the presynaptic terminal, from some sort of precursor. And the precursor can come from anywhere. So sometimes it can be a molecule that's just stored in the cell. Sometimes it's a molecule that's produced by astrocytes or just found in the extracellular fluid. And then they can be taken up into the cell normally these precursors are used to make other things. So sometimes some neurotransmitters are derived from sugar molecules or from other amino acids, and they go through a complex biosynthetic pathway to get to be neurotransmitters. But what's important is that the neuron itself has these special synthetic enzymes. So an enzyme, you may know, is a protein that acts as a catalyst, and it sort of catalyzes biochemical reactions. And so the synthetic enzymes for neurotransmitters are enzymes that specifically catalyze the chemical reactions that turn the precursor into the neurotransmitter. And sometimes there's just one synthetic enzyme, sometimes there can be several. But they're specific to that neurotransmitter and to that neuron. So that means that if you have a neuron that, for example, releases serotonin, then the synthetic enzymes for making serotonin have to be in that neuron, and those synthetic enzymes will not be found in other neurons. In fact, that's one of the ways, we'll talk about this later, but that's how you can identify a type of neuron. If you want to know what kind of neurotransmitter it releases, you can look for those synthetic enzymes. In fact, just as an example, let's take serotonin. So serotonin, it turns out, is made from, its precursor is this amino acid called tryptophan. So tryptophan is an amino acid, even though serotonin itself is not an amino acid. But tryptophan is just one of the 20 amino acids that every cell in your body carries around for making proteins. And so in serotonin neurons, some of that tryptophan just gets converted into serotonin. But first, it has to be converted by this enzyme called tryptophan hydroxylase. Tryptophan hydroxylase takes tryptophan and adds a hydroxyl group to it. That's why it's called tryptophan hydroxylase. And the hydroxylated form of tryptophan is called 5-hydroxytryptamine. That's what this is, 5-HTP. And then a different amino acid, or a different enzyme called aromatic amino acid decarboxylase takes 5-HTP and removes the carboxylic acid group. That's why it's called decarboxylase. That's also why serotonin is not an amino acid, because this enzyme takes that carboxylate group off. And so that's why serotonin also sometimes is called 5-HT, which is short for 5-hydroxytryptamine. And so it is basically the decarboxylated form of 5-hydroxytryptophan, which is, again, sort of the intermediate here. So again, this is the specific pathway for making serotonin that only happens in serotonin neurons. So once you make your neurotransmitters, the next step is to store them into inside of those synaptic vesicles. So that is carried out by specific transporter proteins. In fact, they're specifically called vesicular transporters because their job is specifically to take those neurotransmitters and transport them from the cytosol into the synaptic vesicles. So these vesicles are made of lipid membranes, just like the membrane of the cell itself. So neurotransmitter molecules can't cross in or out on their own. They're too big, they're polar. So just like any other molecule like that, they are kept out of the synaptic vesicle simply because of their physical nature. So these transporter proteins act like pumps, just like the ion pumps that we talked about before. They actively move the neurotransmitter from the cytosol to the inside of the synaptic vesicle. And so in the terminal, then you have these two big populations of vesicles. Most of the vesicles are hanging around just in the cytosol, waiting their turn essentially to be released. And that collection is called the reserve pool. So when a neuron is kind of at rest, most of the neurotransmitters are stored in vesicles that are in the reserve pool. And then eventually the vesicles get moved down to the membrane right next to the place where they're going to be released. And this is the active zone. So the active zone is just the region of membrane where the synaptic vesicles that are ready to be released are attached. They're actually physically attached to the membrane. And there are also a bunch of other proteins that are sort of embedded in the membrane around the active zone that anchor the vesicles and get the process of neurotransmitter release prepared. So next time we will talk about how neurotransmitters actually are released.